Hello, am I audible? Very good evening to the student who have joined. Can you please let me know in the chat box if my voice is audible to you? And also let me know if you can see the presentation and the video clearly. Okay, thank you for confirming. We will be starting at 6 p.m. Till then, please wait. Meanwhile, if you have any doubt or query from the previous class, you can let me know. We will start our today's session at sharply at 6 p.m. So please wait till then. Okay. So very good evening to the students who have joined. I hope my voice is audible and you can see the video clearly. We will wait till 5 more minutes and then we will start our session at 6 p.m. Okay. Meanwhile, if you have any doubt or any query regarding our last classes or any other classes, you can let me know because today it is our last class, okay, last live session. Only tomorrow we will have another class of 1 hour from 4 to 5 p.m. That is a summary class, okay. So if you have any problem regarding any topic from our previous lectures, you can let me know today. Then what I will do is I will cover that topic in the upcoming class, which is scheduled on tomorrow. Tomorrow it will be the last class, last live session. Okay. So if you have any doubt or any query, please let me know beforehand so that I can prepare accordingly for the last class. Let us wait for some time. Then I will start today's class. Let me know if there is any issue with the audio or the video. Okay. We are going to start in a minute, so just wait. Okay, so it is 6 p.m. Let us start our session. Let me first introduce myself. So, very good evening to the students who have joined. My name is Mousumi and I am a PMRF fellow from the Department of Chemistry and Chemical Biology, IIT ISM Dhanbad. I am your TA for this NPTEL SWAM course of Organic Chemistry and Biology and Drug Development, and today it is our week 12 live session. Before starting the session, let me clarify a few things that there will be a problem solving session every week. So till now we have done already 11 problem solving sessions, and these sessions were scheduled on Friday from 8 to 6 p.m. Today it is the last class, our last Friday class, which I am taking right now, and we will have one more class tomorrow, which is from 4 to 5 p.m. Okay, 1 hour summary class that will be. So that is not a regular class. That is basically a summary session when I will summarize the topics from the course, few topics which I think I have not given much importance during our sessions, because that was not very much directly involved in your talk course, but the topics are very important. Few topics I will just try to elaborate so that you can remember the things which we have completed, like very before, I mean earlier in our sessions. So the Google Meet link will be same for the tomorrow's class as well, which you have used for joining today's class. That is this one. Okay. So if any network disruption happens from my side, please do not leave the session immediately. I will try to rejoin as soon as possible. And if there is any problem from your side, you can rejoin at any time. The call is open. So if there is any network issue from your side, you can rejoin back at any time. And I am recording the session. Okay. So because I am recording the session, so that I can record it without any problem, I have muted your microphones and cameras, but the chat box is always open, and I am monitoring the chat box every now and then. So if you have any doubt, any query, anything, you can let me know via the chat box. Okay. Any issue, any problem regarding the voice, the video, or the presentation, or any kind of thing, any doubt, you can let me know through the chat box. The chat box is always open, and I am monitoring the chat box. Since I am recording the session, this recording and this PPT which I'm showing you for during the class. This too I will be sharing with you after the class. Okay, and if there is any doubt, you can let me know via email. Also, you can let me know via the discussion forum. You can let me know via the chat box. The chat box is always open. So, and I'm telling you every now and then, if you have any doubt or any query, please let me know through the chat box. Okay, and as I have mentioned, this presentation and class recording, it will be shared with you after the class. So where you have to find, you have to go to the SWAYAM portal. Okay, you have to go to the NPTEL SWAYAM portal by using your login ID and password or your Gmail. Then after logging in, you will find there is a reminder. So here you can see there is a reminder for tomorrow's problem solving session also. Okay, which is the summary session that I have mentioned you. And then you will find there is a left panel when where there is the weekly course announcement, right? Weekly the course material announcements and all the things are there. In this left panel, here you will be finding a thing which is called the problem solving session 202 July 2025. If you click on this problem solving session recording, then you can directly, you will be redirected to a page which looks like this, which contains every classes, Google Drive link, which will contain every classes, the particular PPTs, and also you will find the class YouTube links of the classes. Okay, so this you will find from your SWAYAM portal. You can download the classes, the PPTs, and you can see the previous classes from the YouTube. Okay. So if you are the email ID, my email ID I will be sharing after the end of the class. So if you have any doubt or any query, you can let me know via email also after ending our classes. Okay. So today it is our week 12 live session. So what we do in our every live session, I show you around 12, around 20 questions. Okay, all questions are MCQ, just like you have in your assignments. Okay, the all question will have four different options. You have to choose any one of the following, A, B, C, or D. Which of the following you think is the correct answer? You have to choose and you have to let me know in the chat box. Okay, I will wait after showing you a particular question. I will wait for some time and then I will check the chat box. Okay, so please try to answer the questions. Even if your answer is wrong, it does not matter, but please try to answer. Okay. So today it is our week 12 live session. So today we are going to discuss about our week 12 course content. In our last class, in our week 11 live session, we have majorly discussed about the polypeptide biosynthesis and antiviral drugs. We have discussed about antiviral drugs today. Antiviral drugs earlier we have discussed about the antibacterial therapeutics in our week 10, I guess. So let us see first what are the week 12 course content. So the week 12 course content is very important, and there are so many things which we are going to cover today. We have the cancer and chemotherapy, we have anti-cancer drugs, we have aromatase inhibition and anti-ulcer drugs, we have cholesterol lowering drugs and cholesterol biosynthesis, and also we are going to discuss a little bit about the QSAR principle, pharmacokinetics and pharmacodynamics. Okay, pharmacokinetics I think I have discussed earlier, and pharmacodynamics little bit today we will discuss. I will discuss this pharmacokinetics and pharmacodynamics topics in more details in our tomorrow's class. Okay, so today is basically we are going to discuss these topics. Now let me go to the first question of today's session. So the first question of today's session says that identify the statement that is incorrect. Your options are: number A, DNA synthesis checkpoint exists in the growth phase of the cell cycle. Number B, in the synthesis phase, a DNA synthesis checkpoint is present. Number C, cancer can result when one or more of the cell cycle checkpoints malfunction. And number D, G2 phase occurs between S and M phase. So which of the following statement is incorrect? Again, it is a correct incorrect kind of question, which I always emphasize that you have to be extra careful because many students make mistakes during this kind of questions. Okay, so read all the options carefully. Then let me know A, B, C, or D, which of the following statement you think is the incorrect one. Let me know via chat box that A, B, C, or D, which of the following statement you think is the incorrect one. Again, I will repeat option number A, DNA synthesis checkpoint exists in the growth phase of the cell cycle. Number B, in the synthesis phase, a DNA synthesis checkpoint is present. Number C, cancer can result when one or more of the cell cycle checkpoints malfunction. And number D, G2 phase occurs between S and M phase. Which of the following statement is incorrect? Let me know A, B, C, or D. Which of the following statement is incorrect? Read the options very carefully and then let me know in the chat box that which of the following statement you think is incorrect. I will wait for a minute or two, then I will check the chat box. DNA synthesis checkpoint exists in the growth phase of the cell. Number A. Number B. In the synthesis phase, a DNA synthesis checkpoint is present. Number C. Cancer can result when one or more cell cycle checkpoints malfunction. Number D. G2 phase occurs between S and M phase. Which statement is the incorrect one? Okay, let me check the chat box now. Okay, I got one answer which is saying that number A is the correct answer, means statement number A is the incorrect one. Okay, let me check if number A is the correct answer for problem number one or not. So the correct answer will be yes, it is number A, which says that DNA synthesis checkpoint exists in the growth phase of the cell cycle. So this statement is incorrect. Other statements are correct. Okay. So now let us understand what are the cell cycle checkpoints. What is the cell cycle and what is the cell cycle checkpoints? So the following diagram describes what different checkpoints in the cell cycle are responsible for. So whenever there is a cell division, so there are different phases. Okay, and in every and there are several checkpoints. So what are the phases? So there are basically major four stages. Okay, one is the G1 stage, then we have a S stage, then is we have the G2, so, and then we have the M stage. Okay, so first is the first phase, which is the G1 phase. This is known as the growth phase 1. Okay, so in the growth phase 1, we have a checkpoint which is known as the cell growth checkpoint. So what is the cell growth checkpoint and what is the, so it occurs at the end of the growth phase 1. At the end of the growth phase 1, as you can see, there is a cell growth checkpoint. It checks whether the cell is big enough and has made the proper proteins for the synthesis phase. If not, the cell goes to the resting phase, which is G0. So there is another phase which is known as the resting period or G0, and there the cell takes rest. If it is not ready for the division, then it will take rest in the resting phase G0 until it is ready to divide. Then we have a synthesis phase or S phase. Okay, so the DNA synthesis checkpoint S is occurs during the synthesis phase. Okay, so it occurs in the synthesis phase or S phase. Here you can see, here we have the DNA synthesis checkpoint in the S phase or the synthesis phase. It checks whether the DNA has been replicated correctly. Okay, so whenever there is a cell, it has a DNA, it has a genetic material and DNA. So it in the synthesis phase, the DNA synthesis checkpoint is checks that whether the DNA has been replicated correctly or not. If so, then the cell continues to the mitosis or M. Then we have a growth 2 phase also, which is the G2 phase. Then we have a mitosis phase or M phase, which is where there is a mitosis checkpoint. What does it do? It checks whether the mitosis is complete. If so, then the cell is the cell divides and the cycle repeats. One important thing is the cancer can result if one or more of these checkpoints malfunction. Okay, so now if you look into our options, the option number A says DNA synthesis checkpoint exists in the growth phase of the cell cycle. It is not true, because we have seen that DNA synthesis checkpoint, it occurs in the synthesis phase, which is the statement number B, which is correct. Then statement number C says cancer can result when one or more of the cell cycle checkpoints malfunction. It is also true. And G2 phase occurs between S and M phase, which is also true. So this is all about your cell cycle and the different checkpoints which are present. So basically, a stalled DNA replication fork will activate in the S phase. So what happens in S phase? A stalled DNA replication fork will activate the S phase checkpoints, where it is ensured a proper replication of the genetic material. Now, if this checkpoint is somehow missed, then other checkpoint at G2 phase reassure proper replication devoid of any DNA damage prior to initiating the mitosis cycle. So here, as you can see, we have the several checkpoints I have mentioned. First, we have a here we have a checkpoint which checks for damaged DNA, here which it checks for the unreplicated or damaged DNA. Here it is also checked for, and at the end of the M phase also there is a checkpoint which checks for the chromosome misalignment. Okay. So this is also a kind of a checkpoint. So is it clear? Is problem number one or the cell cycle and different things are clear? Okay. So I think there is no doubt. Number B was not the was incorrect. Okay. I mean number A was incorrect. But number B was the correct statement because it says that in the synthesis phase we have the I mean the DNA synthesis checkpoint, which is true. Okay. And you have to identify what is the incorrect statement. So which is number A. Okay. So I am going to now the our second question. If there is no doubt. Okay, now let me go to the next problem. So the problem number two, it says that which of the following is responsible for the progression from G1 to S phase? Your options are number A cyclin A and CDK2, number B cyclin B and CDK1, number C cyclin D and CDK4, and number D cyclin E and CDK2. Which of the following is responsible for the progression from G1 to S phase? Your options are number A cyclin A to CDK2, number B cyclin B and CDK1, number C is the cyclin D and CDK4, and number D is the cyclin E and CDK2. Which of the following is responsible for progression from G1 to S phase? Again, I will repeat. Option number A is the cyclin A and CDK2. Number B is the cyclin B and CDK1. Number C is the cyclin D and CDK4. And number D is cyclin E and CDK2. I am going to wait for a minute. Please let me know A, B, C or D. Which of the following you think is the correct answer for problem number 2? Which of the following you think is responsible for the progression from G1 to S phase? See, even if your answer is wrong, it does not matter, but please try. Otherwise, I will not understand that if you are following the class or not. Let me know. A, B, C or D, which of the following is the correct answer for problem number 2? Which of the following is responsible for progression from G1 to S phase? Number A, cyclin A and CDK2. Number B, cyclin B and CDK1. Number C, cyclin D and CDK4. And number D is cyclin E and CDK2. Let me check the chat box. Let me see if there is any answer. Okay, I got an answer which is saying that number C is the correct answer. Let me check if number C is the correct answer or not. Okay. So the correct answer will be, it is number D. Cyclin E and CDK2 are responsible for progression from G1 to S phase. So there are different so the cell cycle actually is controlled by different cyclins and cyclin dependent kinases. So as you can see in our G1 phase we have the CDK4 and 6 and we have the cyclins D1, D1, 2, 3 and the kinases is the CDK4 and 6. At the end we have the cyclin E and CDK2. Okay. Then in the S phase we have the cyclin A and CDK2 and when in the G2 phase we have cyclin A and CDK2 and in the M phase we have the cyclin B and CDK1. So control of the cell cycle involves a variety of proteins which are called cyclins and the enzymes called the cyclin dependent kinase or CDKs. Okay, so CDKs are the family which are the cyclin dependent kinases and cyclin are the proteins which controls the cell cycle. Okay, so there are at least 15 types of cyclin and 9 types of CDK. Each one has a role to play in the different stages of the cell cycle. Examples are shown here in this figure. Okay, binding of an cyclin with its associated kinase activates the enzyme and serves to move the cell from one phase of cell cycle to another. For example, when a cell is in G1 phase, the decision has to be made whether to move to the S phase or the start copying DNA. This decision is depending on the balance of the stimulatory versus inhibitory signal being received through the signal transduction. If the balance is toward the cell growth and division, there is an increase in the cyclin D. Okay, and it binds with the CDK4 and 6. The resulting complexes phosphorylate to a powerful growth inhibitory molecule known as the PRB, which normally binds and inactivates the transcription factor. Phosphorylation alters the PRB such that it can no longer bind to the transcription factor, and the latter is free to bind to specific regions of DNA. This results in the transcription of the specific genes, which leads to the production of proteins capable of moving the cell towards the S phase. Okay, for example, the cyclin E and the thymidine kinase. Once the cyclin E has been produced, it combines with CDK2 and the complex is responsible for progression from G1 to G phase. Okay, so here it is mentioned that once the cyclin E has been produced, it combines with the CDK2, which is the cyclin dependent kinase 2, and this complex is responsible for progressing the cell from G1 to S phase. Other activated cyclin dependent kinase or CDK complexes are important for different phases of the cell cycle. For example, cyclin A CDK2 complex is required for the progression through S phase to S phase, and the cyclin B CDK1 complex necessary for mitosis. As you can see here, all are mentioned that the CDK2 cyclin A complex is necessary for S phase and G2 phase, and CDK cyclin B and CDK1 is responsible for the mitosis, whereas the CDK2 and cyclin E is responsible for progression from G1 to S phase. Okay. So remember, cyclins are the important. So whenever there is cyclin, you have to understand that this is related to our cell cycle and the cell divisions. Okay. Is there any doubt? Do I need to repeat anything? Do I need to repeat about the cyclins, or is it fine? If there is no doubt, then I am going to move to our next problem. Okay, so I think it is fine regarding the cyclins and CDK1s. Okay. If you have any doubt, please let me know. I will repeat at the end of the session also if you have any problem at any question. Okay. Okay, so now let me go to the next problem. Problem number 3 says, what is induced by HIF1 for the survival of the solid tumors? What is induced by HIF1 for the survival of solid tumors? Your options are number A biosynthesis, number B apoptosis, number C angiogenesis, and number D metastasis. What is induced by HIF1 for the survival of solid tumors? Your options are number A biosynthesis, number B apoptosis, number C angiogenesis, and number D metastasis. I will wait for some time. I will wait for a minute, then I will check the chat box. Let me know. What is induced by HIF1 for the survival of solid tumors? Biosynthesis, apoptosis, angiogenesis, or metastasis? I will wait for a minute. Let me know in the chat box A, B, C, or D which of the following is the correct answer for problem number 3. What is induced by HIF1 for the survival of solid tumors? Biosynthesis, apoptosis, angiogenesis, or metastasis? Let me check the chat box. Okay, I got two answers. One is saying number D is the correct answer, and another is saying number C is the correct answer. Let me check which of the following is the correct one, number C or number D. So the correct answer will be, it is number C. Angiogenesis is the correct answer. Angiogenesis is induced by HIF1 for the survival of solid tumors. So what is the angiogenesis? What is angiogenesis? Let me highlight few things. Yeah. So this figure actually shows what is angiogenesis. So as a tumor grows, so whenever there is a tumor, a solid tumor especially, the cancerous cells requires a steady supply of amino acid, nucleic acid bases, carbohydrates, oxygen, and growth factors if they are to continue multiplying. This means the tumor has to have a good blood supply. As the tumor grows in size, however, its cells become increasingly remote from the blood supply and become starved of these resources. Oxygen levels also fall, resulting a state of hypoxia. So whenever there is a deficient of or the deficiency of oxygen, the condition it is known as the hypoxia, right? This is particularly true for the cells that is the center of a solid tumor. As a result, this hypoxia inducible factors, so HIF1 it is known as the hypoxia inducible factors, okay. Sometimes terms will be given to you in like this abbreviated form, okay, so you have to just think and you have to identify what is the full form. So HIF1 means hypoxia inducible factors, okay. It starts to build up within the tumor cells, and these factors serve to upregulate genes that promote survival in the oxygen starved environments. For example, growth factors are released from the cells such as the vascular endothelial growth factor or VEGF and fibroblast growth factor FGF2, which interact with the receptors on the endothelial cells nearby blood vessels and stimulate these cells to divide, and leading to a branching and extension of existing capillaries, a process known as the angiogenesis. Okay. So what is the angiogenesis process? When there is kind of HIF or hypoxia inducible factors are triggered. So what will happen? Growth factors are released from the cell such as the VEGF or vascular endothelial growth factor and FGF2 or fibroblast growth factor, which interact with the receptors of the endothelial cells nearby blood vessels and stimulate these cells to divide, leading to the branching and the extension of the existing capillaries. Okay, so this is angiogenesis. As you can see, this is a tumor cell. So whenever the tumor cell, the cells are become very remote. So what will happen? It will trigger the VEGF and FGF2, which will trigger the endothelial cells, and new blood vessels will form. Okay, and this is known as the angiogenesis. So angiogenesis defined as the formation of new capillaries from the pre-existing vasculature by the migration and proliferation of the endothelial cells. Okay, so this figure also indicates the angiogenesis, the phenomena of angiogenesis. So I hope it is clear what is angiogenesis and how it happens. I think there is no doubt. If there is any doubt, please let me know. Otherwise, I am going to move on to our next problem. Okay, let me go to our next problem. So problem number 4 says, which of the following is not an anthracycline anti-cancer drug? Your options are number A doxorubicin, number B epirubicin, number C darunavir, and number D all of the above. Which of the following is not an anthracycline anti-cancer drug? Doxorubicin, epirubicin, darunavir, or all of the above? I am going to wait for a minute, then I will check the chat box. Let me know A, B, C, or D. Which of the following is the correct answer for problem number 4? Which of the following is not an anthracycline anti-cancer drug? Doxorubicin, epirubicin, darunavir, or all of the above? Which of the following is not an anthracycline anti-cancer drug? Doxorubicin, epirubicin, darunavir, all of the above. If you have paid attention to our in our last class, I think you will correctly answer this question. Okay, let me check the chat box now. Let's see if there is any answer. I have got two answers. One is saying number B, another is saying number C. Let me check which of the following is the correct answer. So, the correct answer will be, it is number C. Darunavir is not an anthracycline anti-cancer drug. Darunavir is a antiviral agent. Okay. In the last class, I have discussed regarding this darunavir a little bit. I have mentioned the name, I guess. I hope so. Darunavir is not an anthracycline anti-cancer drug. It is an antiviral drug. Okay. Doxorubicin, epirubicin, both are the anthracycline anti-cancer drug. So these are the class of the anthracyclines or anthracycline anti-cancer drug. This is the basic structure, as you can see. This is the basic structure. It looks little complicated, but if you practice one or two times, then you will find it is not very much complicated. Okay, so there are planar rings here. Anthracycline system, as we see in the anthracycline. So as you can see, there is a sugar moiety here, and we have different R's here. We have a R1 here. We have a R1 here. We have a R2 here, right? We have a R3 and R4 here. So when the R1, R2, R3 and R4, the changes, the group are changed and the names are changing like this. If R1 is OME, R2 is OH, and R3 is OH and R4 is H. So this is known as doxorubicin. Then we have epirubicin, darunarubicin, and idarubicin. So all are the anthracyclines, and these are the structures R1, R2, R3 and R4. So these anthracyclines are known as the intercalating agents. Okay, so what are the intercalating agents? Let us discuss, because this is a very important thing. Intercalating drugs, they contain a planar, aromatic or heteroaromatic ring system which can slip into the double helix of the DNA. So as you can see, this portion is planar. This blue, the portion which is drawn in blue, this is a planar ring system. Okay, so intercalating drugs are the drugs which contain a planar, aromatic or heteroaromatic ring system which can slip into the double helix of the DNA and distort its structure. Once bound, the drug can inhibit the enzymes involved in the replication and transcription processes. Examples include the dactinomycin and doxorubicin. Doxorubicin, previously known as the adriamycin, belongs to a group of naturally occurring antibiotics called the anthracyclines, which was isolated from the streptomycin pusatius in the 1967. It is a very similar to the structure of the daunorubicin, which is differing only one hydroxyl group. Okay, so as you can see, doxorubicin and daunorubicin both have similar structure, only here R2 is OH and here R2 is H. Otherwise, all are same. Okay. However, this is an important effect of activity, and doxorubicin is one of the most effective anti-cancer agents ever discovered. This is very important. Okay, you have for that doxorubicin is one of the most important anti-cancer agents ever discovered. The drug intercalates into the DNA and is an example of topoisomerase 2 poison. I think I have discussed about the topoisomerase 1, 2 inhibitors and poisons in our mid-hour session, I think in 5th or 6th class, as it is stabilizes the complex formed between the DNA and topoisomerase 2, an enzyme that is crucial for the replication process. It is thought that an excessive number of these stabilized DNA enzyme complexes triggers the apoptosis. Because these enzymes are active during the cell growth and division, the topoisomerase 2 poisons are most effective against the rapidly proliferating cells. A second mechanism by which doxorubicin can prove harmful to DNA involves the hydroxyquinone moiety, which can chelate iron to form the doxorubicin DNA iron complex. Reactive oxygen species are then generated, leading to the single strand breaks in the DNA chain. This mechanism is considered less important than the interaction with the topoisomerase 2, but it has been implicated in the cardiotoxicity of the doxorubicin. A third proposed mechanism involves intercalated doxorubicin inhibiting the helicases which unravel the DNA into the single DNA strands. A variety of other anthracyclines are used in the cancer chemotherapy, mainly the daunorubicin, which is also called the cerubidin, donomycin and rubidomycin. And the second generation anthracycline is the epirubicin and idarubicin or idamycin. Idarubicin lacks the methoxy group at R1. As you can see here, idarubicin does not have methoxy group in the R1, so it is more polar and has altered metabolism, which belongs to its half-life. Third generation of the anthracycline is being studied where ammonium ion, this ammonium ion in the sugar ring is replaced with the azido group or triazole group. Such compounds do not appear to be susceptible to the efflux mechanism, which leads to the drug resistance, but also show decreased general toxicity. Okay. So this is all about the intercalating agents, the mechanism of action of anthracyclines, the examples. Okay. These intercalating agents are very important as anti-cancer drugs also. They are very crucial and they are very important for your syllabus also. So I hope it is you have understood the things. If not, please let me know. Here all the things are mentioned, the mechanism of action, the examples and the structure also. Just you have to go through the slides. Okay. Let me now go to the next problem then. Problem number 5, it says that which of the following is not a third generation aromatase inhibitor? Your options are number A anastrozole, number B letrozole, number C borozole and number D fedrozole. Which of the following is not an third generation aromatase inhibitor? Anastrozole, letrozole, borozole or fedrozole? A, B, C or D, let me know which of the following will be the correct answer. Which of the following is not a third generation aromatase inhibitor? Anastrozole, letrozole, borozole or fedrozole? I will wait for a minute or two, then I will check the chat box. Meanwhile, let me know which of the following is not a third generation aromatase inhibitor? Anastrozole, letrozole, borozole, fedrozole? Which of the following is not an third generation aromatase inhibitor? I will wait for a minute, then I will check the chat box, then we will discuss about the aromatase inhibitors, what is the mechanism of action and what is all about it. Let me check the chat box. Okay, I got two answers. One is saying number A, another is saying number D. Let me check which of the following is the correct answer. So the compound which is not a third generation aromatase inhibitor will be, it is number D, fedrozole. So what is aromatase inhibitors? So aromatase inhibitors, they tend to be used as the second line drugs for the treatment of estrogen dependent breast cancer. Estrogen-dependent breast cancer, a membrane-bound enzyme complex, breast cancer, a membrane-bound enzyme complex consisting of two proteins. One is cytochrome P450 containing heme, and other is the reductase enzyme using NADPH as a cofactor. Aromatase catalyzes the last stage in the last stage in the biosynthesis of the estrogens from androgens, where an aromatic ring is formed. Okay. So let me show you. So this is the reaction which is catalyzed by the aromatase enzyme. So what does it do? It catalyzes the formation of this aromatic ring here. Okay. So basically aromatase catalyzes the last stage in the biosynthesis of estrogen from androgens. This is the androgen X and Y. Androgen XY is the equal to ketone and testosterone. So it is the androsterone if X and Y is the ketone, and it is testosterone if X is OH and Y is H. Okay, where an aromatic ring is formed. As you can see, this aromatic ring is forming here. If it is ketone, this then it is known as the estrone, and it is the OH and H, then it is known as the estradiol. The cytochrome enzyme contains heme, which serves to bind the steroid substrate, and oxygen then catalyze the oxidation. Since the enzyme catalyzes the last step in the synthesis, it has been seen as an important target for the design of the anti-estrogenic drugs. Two types of the inhibitors are used clinically: reversible competitive inhibitors and irreversible inhibitors acting as a suicide substrate. Aminoglucothiamide is an early example of reversible competitive inhibitor, but it has disadvantages in that it binds to various cytochrome P450 enzyme and inhibits a range of the steroid hydroxylations. This results in undesirable side effects. Drug design is based on the aminoglucothione that lead compound can consider a more selective inhibitors, just as a anastrozole and letrozole, which are used to treat breast cancer. The infonitrogen of the triazole, this is the anastrozole, this is the aminoglutamide, the structure, this is the anastrozole, and this is the letrozole. The N4 of the triazole ring, this one, interacts with the heme iron of the aromatase and prevents from binding to the steroid substrate. The anilino-nitrogen of the aminoglutamide serves as the same purpose. Formastine acts as the suicide substrate of the permanently inactivates the aromatase and its action than aminoglutamide. Okay. So these are the generation. The first generation type 1 steroidal inhibitor is none. Type 2 non-steroidal is aminoglutamide. The second generation type 1 steroid inhibitor are formastine, and type 2 non-steroidal inhibitor is fadrozole and rogritazole, and third generation the type 1 is the aromacin or exmestane, and the type 2 non-steroidals are anastrozole, letrozole and vorozole. Okay, so fadrozole, as you can see, is the second generation type 2 non-steroidal aromatase inhibitor, whereas anastrozole, letrozole and vorozole, all three are the third generation type 2 non-steroidal inhibitor. So option number D was the correct answer for problem number 5. I hope it is clear what is the aromatase inhibitor, what does this aromatase do, and what is the role of the aromatase inhibitors. You have the structure is very simple. As you can see, I think the structures for the aromatase inhibitors, the letrozole, anastrozole, and aminoglutamide, all are very simple structures. Okay. So I think you can remember the structures very easily. Just you have to remember this might look some a little bit, complicated, but if you practice one or two times, I think it won't be much complicated. Okay. So let me go to the next problem. Problem number 6 says, which of the following is not a PARP inhibitor? Your options are number A, rucaparib, number C, niraparib, number C, lapatinib, and number D, olaparib. Which of the following is not a PARP inhibitor? Rucaparib, niraparib, lapatinib, or olaparib? Let me know A, B, C, or D. Which of the following will be the correct answer for problem number 6? Which of the following is not a PARP inhibitor? Rucaparib, niraparib, lapatinib, or olaparib? Which of the following is not a PARP inhibitor? Rucaparib, niraparib, lapatinib or olaparib? I will wait for another minute, then I will check the chat box. Then I will discuss what is the PARP inhibitor, what are the examples, and what is the role of the PARP inhibitor. All these are the different enzymes, different drug targets. You have to remember examples, you have to remember the mechanism of action. That is very important. Whatever I am saying today, like the intercalating agents, the aromatase inhibitors and everything, the mechanism of actions are very important, which you have to remember again, along with the examples and so on. Which of the following is not a PARP inhibitor? Let me check the chat box. Let me see if there is any answer. Okay, I got two answer. I think it is for, yeah. One is number B, another is number C I have got. Okay, let me check which of the following is the correct answer. So the correct answer is number C, no, lapatinib, it is not a PARP inhibitor. Okay. So inhibitors of the poly-ADP ribose polymerase. So the PARP, the full form is the poly-ADP ribose polymerase. Okay. So a number of research groups are investigating the poly-ADP ribose polymerase or PARP inhibitors as the potential anti-cancer agents. Although none have them reached through the market, the enzyme repairs single strand breaks in the DNA, so its inhibition can eventually result in the double strand breaks in DNA and cell death. Some tumor cells are likely to be more susceptible to the PARP inhibitors than the normal cells. So this is the structures of the different PARP inhibitors. This is the differential R group conferring the different size and flexibility, as you can see, and this portion is common. As you can, this is the shared benzamide core. As you can see here, we have a shared benzamide core, and this is the portion which is differs, which gives the different size and flexibility, right? So PARP targeted agents. So what are the PARP targeted agents? So the single target therapies like rucaparib, niraparib, talazoparib, pamiparib, and fluzoparib. So the approved drugs, olaparib, this first class inhibitor. The design is the benzophenol derivatives and other derivatives and compounds. Dual target therapies like PARP with other like BCL2, DNA, EGFR, EZH2, HD, ACULK, BLM, and all this PRAG. So a promising synergistic combination of PARP with other anti-tumor targets like all of these, and this design pharmacophore of a PARP inhibitor and pharmacophore of other inhibitor can give rise to the new anti-tumor agents, and this technique is known as the pharmacophoric hybridization. So in recent years, PARP or poly-ADP ribose polymerase have gained significant attention in the cancer treatment. Studies indicate that PARP play a crucial role in DNA repair, maintaining the genomic stability, regulating all regular cell activity, and responding to DNA damage. Consequently, with the discovery and exploration of PARP inhibitors have emerged in cancer treatment. Now, these inhibitors are now used in clinical application for the tumor therapy, particularly for BRCA mutation related cancers. A series of PARPs are now involved in the tumor treatment, with several approved clinical in the cancer therapy. This inhibitor disrupt DNA repair by inhibiting PARP enzyme activity, leading to the cancer cell death, thus offering potential for the tumor therapy. We summarize the basically what happens. Let me tell you what is the role of the PARP inhibitors. Okay, so PARP, as you can see, this it is mentioned that this enzyme repairs a single strand breaks in the DNA, and so the inhibition can eventually result in the double strand DNA breaks and the cell death of the cancer cell. Now the question may arise that why this particular PARP inhibitors, they will not hamper the our normal healthy cell. Okay, if we use this PARP inhibitors to kill the tumor cells, they can may also hamper the growth and the health of the normal cells which are non-tumorous. So basically, what happens that there is a BRCA which is known as a gene, which is a tumor repression genes. Okay, so in the tumor cells, the all the tumor repression genes, they are not present. That is why in the tumor cells, they are for their DNA damage repair, they are mostly dependent on this PARP enzyme. But the normal healthy cell, we have the tumor suppressor genes and proteins. So we the normal healthy cells are not very much dependent on this PARP enzyme like the tumor cells. That is why this PARP inhibitors, they mostly they they are the tumor cells are more susceptible to the PARP inhibitors than the normal cell. This is the reason which is mentioned here. I am highlighting this portion. This is very important thing, that why this cancer cells are susceptible to the PARP inhibitors, not the human cell, because the only the cancer cells are mostly dependent on this PARP for their DNA damage break, not the normal cell. And these are the examples I have mentioned: olaparib, rucaparib, niraparib, and talazoparib. Okay, and lapatinib, which was the option number, I think option number C. So lapatinib is basically a kinase inhibitor. So that is not a PARP inhibitor. So is it clear what is PARP inhibitor? What are the role of the PARP inhibitors and so on? If there is any doubt, I am saying again and again that if there is any doubt, please let me know through the chat box. Okay. Let me go to the next problem if there is no doubt in this PARP inhibitor. So the problem number 7 says, which of the following is not a microtubule inhibitor? Your options are: number A, cisplatin; number B, paclitaxel; number C, vinblastine; number D, colchicine. Which of the following is not a microtubule inhibitor? Cisplatin, paclitaxel, vinblastine, or colchicine? I will wait for a minute, then I will check the chat box. Let me know A, B, C, or D. Which of the following is the correct answer? Which of the following is not a microtubule inhibitor? Cisplatin, paclitaxel, vinblastine, or colchicine? Which of the following is not a microtubule inhibitor? Cisplatin, paclitaxel, vinblastine, or colchicine? Let me know A, B, C, or D. Which of the following you think is the correct answer for problem number 7? Which is not a microtubule inhibitor? Cisplatin, paclitaxel, vinblastine, colchicine? Let me check the chat box now. Let me see if there is any answer for problem number 7. Okay, I got two answers. One is saying number A, another is saying number D. Let me check A or D, which of the following is the correct answer. So the correct answer will be number A, cisplatin. It is not a microtubule inhibitor. So what are the different drugs that are basically targets our tubulin? So we have these drugs, like we have the classes of the drugs which are the microtubule inhibitor. Here we can see from the table we have the paclitaxel. which is the binding domain is the taxane binding. The cancer treatment, breast, ovarian, prostate and lung. And mode of action, antimicrotubule depolymerization leading to the mitotic arrest. We have the docetaxel, which is the taxane binding again. It is used for the treatment of breast cancer, non-small cell lung cancer, androgen independent metastasis prostate cancer. And mode of action is the antimicrotubule depolymerization and the annulation of BCL2 and BCL-XL gene expression. Colchicine is the colchicine binding, which is the hepatocellular and prostate cancer. It is used. Antimicrotubule polymerization, cell cycle arrest in the metaphase, and vinblastine is the vinca binding. Testicular Hodgkin's and non-Hodgkin's lymphoma, breast and germ cell cancers induces the wedge at the tubulin interface, causing tubulin self association with the spiral aggregates. Anti-microtubule polymerization and cell cycle arrest. So these are the drugs that target the tubulin. So we have the vinca alkaloid like vincristine and vinblastine, which binds with the tubulin, inhibit the microtubule formation, metaphase arrest, and it shows the anti-cancer effect, right? We have the taxanes. Taxanes are the anti-cancer, and they freezes the microtubule in the metaphase. Okay, then fuses the microtubule in metaphase and tubulin polymerization fails to depolymerize. Then we have colchicine, which binds to the tubulin, inhibit the microtubule depolymerization, impair activation and migration, and reduce inflammation. Now this is used for the neutrophils. Okay. Then we have the griseofulvin, which binds to the fungal tubulin, impaired microtubule formation, inhibit, and this is the fangistatic. Then we have this mebendazole and albendazole, which blocks the polymerization of tubulin in the intestinal cells, blocking the glucose and glucose uptake and the death of the hermanthus. So these are the different drugs that target tubulin. So paclitaxel, vinblastine, colchicines, all are the microtubule inhibitor except cisplatin. Cisplatin, the mechanism of action of anti-cancer effect of cisplatin is different. That basically they cross-links with the DNA, and the complete mechanism is different. It is not a microtubule inhibitor. So number A was the correct answer. So is it clear? Problem number 7. If there is any doubt, let me know. Otherwise, I will move to the next problem. If you have any doubt or any query at any question, just write in the chat box. At the end of the session, I will discuss that particular question. Okay. Let me go to the next problem. Okay, the mechanism of action of the microtubule inhibitors. The main mechanism of the cytotoxic action involves binding to the cisplatin. So this is a cisplatin, the mechanism of action of cisplatin. The mechanism of action of cytotoxic action involves binding to cisplatin to genomic DNA in the cell nucleus to form intrastant and intrastant crosslinks. This interferes with the normal transcription or DNA replication mechanisms and triggers cytotoxic processes that lead to the cell death. So problem number 8 says which toxic side product is generated from the alkylating agent cyclophosphoramide. Your options are number A acrolein, number B protonaldehyde, number C cytosine and number D guanine. Which toxic side product is generated from the alkylating agent cyclophosphoramide? Acrolein, protonaldehyde, cytosine or guanine. Again I will wait for a minute, then I will check the chat box. Okay. Let me know which of the following toxic side product is generated from the alkylating agent cyclophosphamide: acrolein, protonaldehyde, cytosine, and guanine. Which toxic side product is generated from the alkylating agent cyclophosphamide? Acrolein, protonaldehyde, cytosine, or guanine? I'm going to wait for a minute, then I will check the chat box. Which toxic side product is generated from the alkylating agent cyclophosphoramide? Let me check the chat box now. Let me see if there is any answer for problem number 8. Okay, I got an answer saying number A is the correct answer. Both are saying. Okay, let me check if number A is the correct answer or there is another correct answer. So the toxic side product which is generated from the alkylating agent cyclophosphoramide will be very good. It is acrolein. So this is cyclophosphoramide. As you can see, this is the structure of cyclophosphoramide. So it is the reaction which is the generation of the phosphoramide mustard from the cyclophosphoramide. So first we have, this is the structure of cyclophosphoramide by the enzyme cytochrome P450 enzyme. It will create, it will oxidize in this position, and then this oxygen lone pair gives here, and we have this kind of ring opening which gives us the cytotoxic alkylating agent which is known as the phosphoramide mustard, and the side product is acrolein, which is toxic again. So cyclophosphoramide is the most commonly used alkylating agent in cancer chemotherapy. It is a non-toxic prodrug which is converted to body to its active drug. This is important to remember, that it is a non-toxic prodrug which is converted in the body to the active drug. Metabolism takes place in the liver where cytochrome P450 enzymes oxidize the ring. The ring opening then takes place and non-enzymatic hydrolysis splits the acrolein from the molecule to generate the cytotoxic alkylating agent. The nucleophilicity of the nitrogen is tempered by being part of the phosphoramide group, and so the active agent is more selective for the stronger nucleophiles such as the guanine. Cyclophosphoramide itself is relatively non-toxic and can be taken orally without causing damage to the gut wall. It was also hoped that the high level of phosphoramidase enzyme present in some tumor cells would lead to the greater concentration of the alkylating agent in these cells and the result in some selectivity of action. Unfortunately, the acrolein released can sometimes prove toxic to the kidneys and bladder. One possible explanation is the acrolein alkylates with the cytosine residue of the cell proteins. Certainly, the toxicity can be reduced by co-administrating the sulfhydryl donors such as the N-acetylcytosine or the sodium 2-mercaptane sulfonate or mesna to interact with the acrolein. So this is the how the toxicity of acrolein can be neutralized. So we have to co-administer this particular cyclophosphoramide with these agents which can interact with acrolein and reduce the toxicity. Okay. So this is to very remember important, what the cyclophosphoramide, what is the mechanism of action of this alkylating agent, this reaction, and also why the acrolein is toxic and how to tackle the toxicity of acrolein. Okay, I hope it is clear. If not, please let me know in the chat box. I will go to the next problem. So if you have any doubt or any query, let me know. Okay. Let us go to the next problem then. The problem number 9 says, which of the following statement is untrue regarding mitomycin? Your options are: number A, it is an excellent choice of drug for solid tumors. Number B, it is activated by the bio-oxidative process. Number C, it is a prodrug. And number D, it is a mechanism of action when there is a Michael reaction. Which of the following statement is untrue? Which of the following statement is untrue regarding mitomycin? Number A, it is an excellent choice of drugs for the solid tumors. Number B, it is activated by bio-oxidative process. Number C, it is a prodrug. And number D, in its mechanism of action, there is a Michael reaction. Let me know A, B, C, or D. Which of the following statements is untrue regarding mitomycin? I will wait for 2 minutes. Please read the options carefully, then let me know. Which of the following statements is untrue regarding mitomycin? Number A, it is an excellent choice of the drug for solid tumors. Number B, it is activated by bio-oxidative process. Number C, it is a prodrug. Number D, in its mechanism of action, there is a Michael reaction. Which of the following statements is untrue regarding mitomycin? Number A, it is an excellent choice of drug for solid tumors. Number B, it is activated by bio-oxidative process. Number C, it is prodrug. Number D, in its mechanism of action, there is a Michael reaction. Let me check the chat box now. Let's see if there is any answer. Okay, I got an answer. Number B and number C. So I got two answers. One is number B, another is number C. Now let me check number B or number C. Which of the following is the correct answer for problem number 9? The correct answer is, it is number B. It is activated by the bio-oxidative process. This statement is untrue. Okay, so this statement is not correct. So this is the structure of the mitomycin. First, as you can see, there is a quinone type of system, which is first is reduced, okay, and then followed by the elimination of methanol from this position. So then we will have a double bond here. Then what will happen? This oxygen lone pair gives it here, and there is a bond rotation which opens this ring, and subsequently which this ring opening will lead to this kind of alkylating this compound, which is an alkylating agent. Okay. Then DNA, we have free NH2 group which reacts with this ring, and we have this kind of structure. So this is the cross-linking with the DNA. Okay. So the major class of the drug is mitomycin. Mitomycin was discovered in 1950s, and again it is being isolated from the class of microorganism known as the streptomyces. It is one of the most toxic anti-cancer drugs in the clinical use, and it is another example of the prodrug. Of course, so we will look into the prodrugs much more later in the course. Okay, so which happens, the enzymes which carry out which is the known as the bioreductive process. So this is the activation. This mitomycin is a prodrug. Okay, which is true, and the activation, it is not an bio-oxidative process. This is a bioreductive process. There is many more enzymes. As you can see, the first step is the reduction step, right? So there are many enzymes in our body which can do oxidation as well as reduction, and they are known as the oxidoreductase. So when a mitomycin encounters a bioreductive enzyme, this quinone undergoes a reduction to form a diol, and now quinone is actually electron withdrawing group. We will imagine the carbonyl groups as the electron withdrawing, whereas the diol is fairly electron rich, which can promote the rearrangement reaction, which eventually prick the proton and produces the species and becomes the substrate for attack the DNA. As the reduction step is involved in mechanism, it has been proposed that this drug should be more effective against tumors in a oxidant starved or hypoxic environment such as the solid in the center of the solid tumor. So as you can see, the first step is the bioreductive process, means reduction is happening. So since the reduction is happening, which means that this drug will be suitable for the tumors which have this oxygen less environment or the hypoxic environment, which are the solid masses or solid tumors. Okay. So now let us look at the statements. The first statement says it is an excellent choice of drug for solid tumor, which is true. It is activated by biooxidative process. It is not true. It is activated by bioreductive process. It is a prodrug. It is also true. And its mechanism of action, there is a Michael reaction, which is true. As you can see, this kind of electron flow and this kind of reaction, these are known as the Michael reaction. Okay. So this was the problem number 9. Now I will go to the next problem. The problem number 10 say anti-cancer drug calcimicin is a dash and its mechanism of action contains dash reaction. You have to fill in the blanks. Your options are number A, topo 2 inhibitor and Bally-Silman reaction. Number B is topo 1 inhibitor and Suzuki coupling reaction. Number C, kinase inhibitor and Diels-Alder reaction. Number D, chain cutter and Boltzmann cyclization. Anti-cancer drug calcimicin is dash. The which type of drug it is? Is it a topo 2 inhibitor, topo 1 inhibitor, kinase inhibitor, or chain cutter? Or its mechanism of action contains which reaction? Bally-Silman, Suzuki, Diels-Alder, or Boltzmann cyclization? You have to fill in the blanks. Again, I will give you 2 minutes, then I will check the chat box. Let me know A, B, C, or D, which of the following will be the correct answer for problem number 10. Anti-cancer drug calcimicin is dash and its mechanism of action contains dash reaction. Which type of this agent it is and what is the mechanism of action contains which reaction? I will wait for 2 minutes, then I will check the chat box. Anti-cancer drug calcimicin is dash and its mechanism of action contains dash reaction. Your options are number A, topo 2 inhibitor and Bally-Silman reaction. Number B, topo 1 inhibitor and Suzuki coupling reaction. Number C, kinase inhibitor and Diels-Alder reaction. Number D, chain cutter and Boltzmann cyclization reaction. What will be the correct answer for problem number 10? Now let me, I think there is the 2 minutes is over. Let me check the chat box. Let's see if there is any answer for problem number 10. Okay, I got two answers. One is saying number D, another is saying number B. Let me check which of the following is the correct answer, number D or number B, or is there any other correct answer for problem number 10? So the correct answer will be, okay, see it is number D, but I just forgot to write the complete term. Just let me. So this is a chain cutter, okay. This is a chain cutter and the mechanism of action, it involves the Boltzmann cyclization reaction. Okay, just let me do it. Yeah. So yes, number D is the correct answer. Anti-cancer drug calcimicin is a chain cutter and its mechanism of action contains the Boltzmann cyclization reaction. Now let us discuss it. So let us first see what is the Boltzmann cyclization. So in case of Boltzmann cyclization, we have a N-diene. So N-diene means there is a triple bond followed by a single bond, double bond. then single bond, then we have another triple bond. So two triple bonds and both are connected with a double bond with a single bond. So this is a known as the indiene. In means a double bond and diene means two triple bonds. Okay. So whenever there is a this kind of structure, if we give it heat or there is a light photochemical reaction or photochemical or any kind of thermal indicate thermal condition is provided, what will happen? 1,2 so that the radical will form. This 1,4 benzodienoid or or parabenzene diradical is formed. Then this diradical, what it will do is it will take up a hydrogen from the nearby molecule like this. Okay, and then we will have this kind of a benzene and this ring. Okay. So this is the mechanism of the Boltzmann cyclization, the cyclization reaction. From this to this is known as the Boltzmann cyclization. What is the mechanism of action of calcimicine? As you can see in the this is the structure of calcimicine. So if there is a this kind of structure, when is there is a nucleophile attacks here and we have a free thiol group here. This thiol group attacks on this position and we have this kind of electron. The electron, the bond rotation and electron gives on to the there to give a O minus, and then what will happen? So as you can see, there is a indiene in this backbone. This indiene will form a 1,4 diradical here. Okay, and this 1,4 diradical, what it will do is it will interact with DNA and form the DNA diradical, and DNA double strand cleavage will happen. So these are known as the chain cutters. So chain cutters cut the DNA strand and prevent the enzyme DNA ligase from repairing the damage. They appear to act by creating diradical on the DNA structure. These diradical react with the oxygen to form the peroxy species and the DNA chain fragments. Calcimicin is an example of the chain cutter. This compound binds to the minor group of the DNA and cuts the DNA chain. The reaction starts with a nucleophile attacking the trisulfide group. The thiol is then fed the underground intermolecular Michael addition and a reactive alpha beta with an reactive alpha beta unsaturated ketone. The resulting intermediate then cycloaromatizes a reaction known as the Boltzmann cyclization to produce an aromatic diradical species that snatches two hydrogen from DNA. As a result, the DNA becomes diradical and the reaction with oxygen leads to the chain cutting. So this is the complete mechanism of action of the chain cutters. So the important thing which you have to remember, that the thiol, which is first the reaction start with the nucleophile which generates a thiol. This thiol is undergoes an intramolecular Michael addition with a reactive alpha beta unsaturated ketone. This step, okay. Then the resulting intermediate, this cycloaromatizes via the Boltzmann cyclization to produce the aromatic diradical species that snatches two hydrogens from DNA. And as a result, the DNA becomes diradical and the reaction with the oxygen leads to the chain cutting. So this is the mechanism of action of the chain cutters, which are a very important class of the anti-cancer drugs. Okay, calcimicin is the example, and this is the mechanism of action. So is it clear or is there any doubt? The mechanism of action of chain cutters, is it clear? If there is no doubt, then I will move to the next problem. As I am mentioning you each and every, I am just trying to giving you each and every example, one example at least of every for all the anti-cancer class of compounds like chain cutters we have discussed. We have discussed in the last question about the mitomycin. Okay, then we have discussed about the alkylating agents also. So all are these are the classes of anti-cancer drugs. I am going to I am just trying to give you an example and discuss the mechanism of action of the each class. Is there any doubt? Otherwise, I will go to the next problem. Let me know if there is any doubt. I will repeat that particular point. Okay. Problem number 11 says camptothecin works as dash. Your options are number A topoisomerase 1 inhibitor, number B topoisomerase 2 inhibitor, number C DNA guidage inhibitor, and number D DNA polymerase inhibitor. Camptothecin works as which kind of inhibitor? Number A topoisomerase 1 inhibitor, number B topoisomerase 2 inhibitor, number C DNA guidase inhibitor, and number D DNA polymerase inhibitor. Camptothecin works as what? Topoisomerase 1 inhibitor, topoisomerase 2 inhibitor, DNA guidase inhibitor, DNA polymerase inhibitor. I will again wait for a minute or two, then I will check the chat box. Let me know A, B, C or D, which of the following is the correct answer. Camptothecin works as which kind of inhibitor? Topoisomerase 1, topoisomerase 2, DNA guidase or DNA polymerase? Camptothecin works as what kind of inhibitor? Let me check the chat box. Let me see if there is any answer. Okay, I got two answers. One is saying number D, another is saying number B. Let me check which of the following is the correct answer, B or D, or is there any other answer? So the correct answer will be, it is A. It is a topoisomerase 1 inhibitor. Okay, camptothecin works as a topoisomerase 1 inhibitor. So camptothecin is a plant alkaloid. Okay, so it is a most established inhibitor of DNA topoisomerase 1. This is a very important. Okay, camptothecin is most established inhibitor of DNA topoisomerase 1 and it is a very, very important anti-cancer drug. So this is the structure of camptothecin in a active form, which have a lactone, and in the carboxylate form it is an inactive form. Okay, topoisomerase 1 is only target of this drug, and CPT binds neither to topoisomerase alone nor to DNA alone, but only to DNA topoisomerase cleavage complex. This is also important. How this why this camptothecin is important and how this is unique, because it does not this is the only target. This is a monotarget drug. Okay, its only target is topoisomerase 1, and it binds neither to topoisomerase 1 alone nor to DNA alone, but to the topoisomerase 1 DNA cleavage complex. So the important molecule we will look into, camptothecin is a natural product that is extracted from the Chinese bush of Camptothecina acuminata in 1966. Camptothecin stabilizes the cleavage complex, and it forms cleavage complex that is formed between the DNA and the enzyme topoisomerase 1. So by doing this, what happens? It results in the single strand breaks that accumulate the DNA. You may remember topoisomerase 1 and 2 both need the DNA for their function. The difference between topoisomerase 1 and 2 is that topoisomerase 1 cleaves only one strand, whereas topoisomerase 2 cleaves both strands of the DNA. Since the complex between DNA and enzyme topoisomerase 1 is stabilized by single strand break persist, it does not therefore accumulate the DNA, resulting the inhibition and the proliferation. Okay, so this is about the camptothecin. Now let me go to the next problem. Okay, the single strand breaks that can repair if the drug repairs, but if the replication is taking place when the drug enzyme DNA complex is present, then irreversible double strand break takes place, which leads to the death. Semisynthetic analogs of camptothecin has been developed clinically as a clinically useful anti-cancer drug. Okay, so this is all about the camptothecin, the mechanism of action, the structure, and why it is important. Okay. Now let me go to the next problem. Problem number 12 says, what is the mode of action of proflavin? Your options are number A, alkylating agent, number B, chain cutting agent, number C, antisense agent, and number D, intercalating agents. What is the mode of action of proflavin? Alkylating agent, chain cutting agent, antisense agent, or intercalating agents? Let me know in the chat box A, B, C, or D, which of the following you think is the correct answer. What is the mode of action for proflavin? Alkylating, is it a alkylating agent or chain cutting agent or antisense agent or intercalating agent? I will wait for a minute or two, then I will check the chat box. What is the mode of action for proflavin? What type of agent is it? It is alkylating agent, chain cutting agent, antisense agent, or intercalating agent. What is the mode of action for proflavin? What kind of agent is proflavin? And alkylating, chain cutting, antisense, or intercalating? I will wait for another minute, then I will check the chat box. Let me check the chat box now. Okay, I got two answer. Both one is saying number B, another is saying number D. Let me check. Number B or D, what is the correct answer, or is there any another answer? So the correct answer will be, it is number D. Proflavin is the intercalating agent. So this is the structure of proflavin. Proflavin is a topical antiseptic agent used for the wound dressing and to prevent infections. So the drugs acting on DNA intercalating agents like proflavin, it basically it actually they slip between this. This has a planar structure. So these are the similar to the we have discussed like anthracyclines, right? Like doxorubicin. So all the this also have a planar structure which slips between these base pairs. Okay, and this planar structure this interacts with the sugar back. This interacts with the base pair. The base pair the planar structures with the van der Waal interaction, and this in the at the side we have this ammonium ions which interacts with the sugar backbone which contains an O negative which interacts with this positive structure. So this is the ionic interaction. Okay. So intercalating agents that contain, so I have discussed it earlier also, that intercalating agents they contain flat planar structure that intercalate themselves into the double helix. This distort the helical structure. At replication, the template contains the intercalated agent which cause the insertion of the random extra base. The base pair addition is complete after another round of replication during which the intercalating agent is lost. Example is the ethereum bromide, proflavin and acridine orange. These are the example of the intercalating agents. So intercalating drugs are compounds that contain a planar or heteroaromatic feature which slip between the base pair layers of the DNA double helix. Some of the drugs prefer to approach the helix via the major group, other prefers via the minor group. Once they are inserted between the nucleic acid base pair, the aromatic or heteroaromatic ring are held by van der Waal interaction with the base pairs above and below. Several intercalating drugs also contain ionized group which can interact with the charged phosphate group of the DNA backbone, thus strengthening the interaction. Proflavin is an example of the group of antibacterial compound called the aminoacridines. Okay. Proflavin is an example of the group of the antibacterial agent compounds called aminoacridine, which were used during the World War I and II to treat deep surface wounds. These drugs proved highly effective in preventing infection and reduced the number of the fatalities resulting from the wound infection. Proflavin is completely ionized at pH 7 and interacts directly with the bacterial DNA. The flat tricyclic ring intercalates between the DNA base pair interacting with them by the van der Waal forces, where the ammonium cations form the ionic bonds with the negatively charged phosphate groups and the sugar phosphate backbone. Once inserted, proflavin deforms the DNA double helix and prevents the normal functions of the replication and transcription. Okay, so this is the mechanism of action for the proflavin. Now I will go to the next problem. Problem number 13 says, which of the following is nitrosourea alkylating agent? Your options are number A, lomustine, number B, carmustine, number C, streptozotocin, and number D, all of the above. Which of the following is nitrosourea alkylating agent? Lomustine, carmustine, streptozotocin, or all of the above? Which of the following is nitrosourea alkylating agents? Again, I will wait for a minute or two, then I will check the chat box. Meanwhile, let me know which of the following is nitrosourea alkylating agent: lomustine, carmustine, streptozotocin, or all of the above. In the chat box, let me know. Which of the following is nitrosourea alkylating agent: lomustine, carmustine, streptozotocin, or all of the above? Which of the following is nitrosourea alkylating agent? Lomustine, carmustine, streptozotocin, or all of the above? Let me check the chat box. Okay, I got one answer saying number C. Let me check if number C is the correct answer or not. So if there is any doubt from the previous questions, please let me know. Okay, I have not asked it separately, but if you have any doubt or query, please let me know. So the correct answer for problem number 13 will be, it is number D, all of the above. Lomustine, carmustine, streptozotocin, all of the above are the nitrosourea alkylating agent. So nitrosourea, the anti-cancer agents lomustine, carmustine were discovered in 1960 are the chloroethyl nitrosoureas which decompose spontaneously in the body to form two active compounds, one alkylating agent and a carbomethylating agent. The organic isocyanate which is form carbomolates the lysine residues in the proteins and may inactivate the DNA reactive enzymes. The alkylating agent which react initially with the guanine moiety on one strand of DNA is then guanine or cytosine unit on the other strand of the to produce the intrastrand cross-linking. Streptozotocin is naturally occurring nitrosourea isolated from the Streptomyces acromonogenes. Okay, so this is the mechanism of actions for the nitrosourea. Okay, this is this mechanism of action. This is the structure of lomustine, this is the structure of carmustine, and this is the structure of streptozotocin. So as you can see, the structure it forms it forms isocyanide and alkylating agent. This alkylating agent basically alkylates with DNA and acts and then there is a carbomolylation also happens with the lysine residue. Okay. And this is the alkylation sites on the guanine and cytosine for the nitrosourea. This is this position. The alkylation happens with nitrosourea on the guanine and on cytosine. In this position, the N3 position, the alkylation happens with the nitrosourea. Okay. So the mechanism of action of the alkylating agents is same what happens with the cyclophosphoramide and other alkylating agents. Okay. So this was the problem number 30. Now I will go to the next problem. Problem number 14 says which of the following enzyme catalyzes the conversion of 3-hydroxy 3-methyl glutarate CoA or to mevalonic acid in the route of the cholesterol biosynthesis. Your options are number A HMG CoA reductase, number B squalene synthase, number C HMG CoA synthase, and number D acyl CoA thiolase. Which of the following enzyme catalyzes the conversion of the 3-hydroxy-3-methyl-glutaryl-CoA to mevalonic acid in the route of the cholesterol biosynthesis? Number A, HMG-CoA reductase, number B, squalene synthase, number C, HMG-CoA synthase, and number D, acyl-CoA thiolase. Let me know A, B, C or D, which of the following will be the correct answer for problem number 14. Which of the following enzyme catalyzes the conversion of 3-hydroxy-3-methyl-glutaryl-CoA to mevalonic acid in the route of the cholesterol biosynthesis? Your options are number A, HMG-CoA reductase, number B, squalene synthase, number C, HMG-CoA synthase, and number D, acyl-CoA thiolase. I will wait for a minute, then I will check the chat box. Let me know. Which of the following enzyme catalyzes the conversion of 3-hydroxy-3-methyl-glutaryl-CoA to mevalonic acid? Let me check the chat box. Let me see if there is any answer for problem number 14. Okay, I got two answers. Both are saying number A is the correct answer. Now let me check if number A is correct answer or is there any other correct answer. So the correct answer is yes, it is number A, HMG-CoA reductase. Okay. So this is the route of the cholesterol biosynthesis which start from the acetyl-CoA, then it goes to the acetoacetyl-CoA by acetoacetyl-CoA thiolase is the enzyme. Then acetoacetyl-CoA goes to the 3-hydroxy-3-methyl-glutaryl-CoA or HMG-CoA. Okay, so and the enzyme HMG-CoA reductase converts this HMG-CoA to mevalonic acid. Then mevalonic acid forms the farnesyl diphosphate which goes to squalene by squalene synthase, and in the during the last phase, the 7-dehydrocholesterol is converted to cholesterol by this 7-dehydrocholesterol reductase. Okay, so this is the step. This is the cholesterol biosynthesis. Several different steps and different enzymes are involved here. Very important is the HMG-CoA reductase. Okay, which catalyzes conversion of the HMG-CoA to mevalonic acid. The HMG-CoA reductase, it is critical rate limiting enzyme in the mevalonate pathway. Its function is to catalyze the conversion of HMG-CoA to mevalonate because it controls the rate of the entire cholesterol production process. Its primary target for the regulation. The HMG-CoA reductase activity is controlled by multiple factors. When cholesterol levels are high, the enzyme's transcription, translation, stability are suppressed to reduce new cholesterol production. Statins, the cholesterol lowering drugs known as statins, work by inhibiting HMG-CoA reductase, which is effectively reduces body's cholesterol synthesis. Okay. Yes, the correct answer was number D. I will go to the next problem. Problem number 15 says statins work as number A, HMG-CoA synthase inhibitor, number B, HMG-CoA reductase inhibitor, number C, squalene synthase inhibitor, number D, acetoacetyl-CoA thiolase inhibitor. Which of the following will be the correct answer? Statins work as what? Number A, HMG-CoA synthase inhibitor, number B, HMG-CoA reductase inhibitor, number C, squalene synthase inhibitor, and number D, acetoacetyl-CoA thiolase inhibitor. I will wait for a minute, then I will check the chat box. Let me know A, B, C or D, which of the following will be the correct answer. Statins work as which kind of inhibitor? Number A, HMG-CoA synthase inhibitor. Number B, HMG-CoA reductase inhibitor. Number C, squalene synthase inhibitor. Number D, acetoacetyl CoA thiolase inhibitor. What will be the correct answer for problem number 15? Let me check the chat box. Okay, I got two answers. One is number B, another is number C. Let me check which of the following will be the correct answer. So the correct answer will be, it is number B, HMG-CoA reductase inhibitor. So HMG-CoA reductase inhibitors are also known as the statins. Okay, they are the class of the lipid lowering drugs that reduce the illness and mortality in those who are at a high risk of the cardiovascular disease. They are the most common cholesterol lowering drugs. They reduce the risk of the illness related to the arteriosclerosis, which the varying degree of the effect on depending on the underlying risk factors and the history of cardiovascular disease. Clinical practice guidelines generally recommend people to start with lifestyle modification through the cholesterol lowering diet and physical exercise. What is the mechanism? The HMG-CoA reductase pathway is blocked by the statins by inhibiting the rate limiting enzyme. Now, the rate limiting enzyme HMG-CoA reductase. Statins act by competitively inhibiting HMG-CoA in reductase. So it is important that statins are the competitive inhibitor for the HMG-CoA reductase enzyme and the rate limiting enzyme of the mevalonate pathway. Because the statins are similar structure of HMG-CoA on the molecular level, they will fit into enzyme's active site and compete to the native substrate, which is HMG-CoA. This competition reduces the rate of the by which the HMG-CoA reduction is able to produce mevalonate. The next molecule in this cascade will eventually produce the next molecule in the cascade that eventually produces cholesterol. So statins, they act as a competitive substrate and they actually fits very well into the active site of HMG-CoA reductase enzyme because of their structural similarity with the HMG-CoA. That is why the taking up the HMG-CoA by this enzyme is diminishing, which will diminish the synthesis of the mevalonate, which actually synthesizes the cholesterol. Okay, so these are the statins which are in market. So like pravastatin, simvastatin, flavastatin. Atorvastatin is a very important drug. Okay, it is very marketed very much. Okay, and used on a very large scale. Rosuvastatin also, pitavastatin, mevastatin, lovastatin. Okay, the green ones are the FDA approval, which year it got everywhere approval, and this red one is the pharmacophore group. Okay. So this was all about the statins. Now let me go to the next problem. Problem number 16 says anti-ulcer drug omeprazole is number A, reversible protein pump inhibitor, number B, antagonist of H plus K plus ATPase, number C, agonist of H plus K plus ATPase, number D, irreversible proton pump inhibitor. Anti-ulcer drug omeprazole is which of the following? Number A, reversible proton pump inhibitor. Number B, antagonist of H plus K plus ATPase. Number C, agonist of H plus K plus ATPase. Or number D, irreversible proton pump inhibitor. Which of the following will be the correct answer for problem number 16? Let me know. Again, I will wait for a minute. We don't have much time. We have to complete 5 more questions. So I will be quick from now on. Anti-ulcer drug omeprazole is which kind of drug? Which kind of therapeutic agent is it? Reversible proton pump inhibitor, antagonist of H plus K plus ATPase, agonist of H plus K plus ATPase, or irreversible proton pump inhibitor? Anti-ulcer drug omeprazole is which kind of agent? Let me check the chat box. Let's see if there is any answer for problem number 16. Got two answer. Number B, both are saying that number B is the correct answer for problem number 16. Let me check if number B is the correct answer or not. So, omeprazole is number D is the correct answer. It is the irreversible proton pump inhibitor. Okay. So this is the structure of omeprazole. As you can see, we have a benzimidazole ring here, and also we have a pyridine ring in its structure. So two heterocyclic rings are present. So the proton pump is only present in the canalicular membrane of the peritoneal cells and is crucial for the mechanism by which hydrochloric acid is released into the stomach. It is the H plus K plus ATPase because it is called an H plus K plus ATPase because it pumps the protons out of the cell into the canaliculus. At the same time, it pumps potassium ions back in. Okay, energy is required for this process as both protons and the potassium ions are being moved against their concentration gradient. In fact, the ratio of the protons inside the cell and the protons on the canaliculus is 1 to 10 to the power 16. The energy is required to carry out this exchange is obtained by the hydrolysis of ATP. That is why the term ATPase comes from. The pump is not responsible for the efflux of the chloride ion. This departs the cell through the separate chloride ion channels. This outflow closely matches with the efflux of the protons, such as the chloride ion is released for the every proton is pumped out. As a result, hydrochloric acid is formed in the canaliculus rather than the inside of the peritoneal cell. As each chloride ion departs the cell, it is accompanied by the potassium ion which flows through its own ion channel. No energy is required for this outflow because the potassium ion is flowing down to the concentration gradient. The potassium ion acts as a counter anion to the chloride ion, and once in the canaliculus, it pumped back to the cell by the proton pump and described previously. Consequently, potassium ions undergo the cyclic movement in and out the cell. So this is the whatever I have mentioned here. This is the role of the proton pump in the secreting hydrochloric acid. As you can see, that this is the proton is coming in at this side in the canaliculus, and potassium is going inside. Okay, and ATP is converted to ADP and PI, which gives us the energy for carrying out this portion. And here chloride ion comes out from the different chloride ion cell that forms the HCl. So this is the lumen of the stomach. Okay, this blue one, this is the proton pump. Okay, and these are the ion channels by which potassium and chloride ions comes out, and this is the receptors. So the proton pump inhibitor, there are four proton pump inhibitors in the clinical use. This is important. Okay, four proton pump inhibitors are in the clinical use. We have the omeprazole, we have lansoprazole, we have pantoprazole, and rabeprazole. So the S enantiomer of omeprazole has also been approved recently. All the proton pump inhibitors have pyridyl methyl sulfonyl benzimidazole skeleton. We have a pyridyl methyl sulfonyl benzimidazole skeleton and acts as a prodrug. Since they are activated when they act as a prodrug, since they are activated when they reach to the acidic canaliculi of the peritoneal cells. Once activated, they bind irreversibly to expose the cytosine residues of the proton pump and block the pump, preventing further release of the hydrochloric acid. The big strategic advantage in inhibiting the proton pump is rather than blocking the histamine and cholinergic receptors. For example, H2 antagonists block the histamine receptors and block the stimulatory effect on the histamine, but it does not block the receptors for the acetylcholine or gastrin. So it is possible to the parietal cell to be activated toward the secretion. The proton pump is downstream of these targets and operates the final step of hydrochloric acid release. Blocking it prevents the release of the hydrochloric acid regardless of what mechanisms are involved in stimulating the hydrochloric acid secretion. Okay. So this is the mechanism of action for the proton pumps and how this works. Now let me go to the next problem. Problem number 17. It says which of the following activities occur following the stimulation of H2 receptor? Your options are number A, vasodilation, number B, uterine contraction, number C, bronchial smooth muscle contraction, number D, enhanced secretion of hydrochloric acid in stomach. Which of the following activities occurred following the stimulation of H2 receptors? Your options are number A, vasodilation, number B, uterine contraction, number C, bronchial smooth muscle contraction, and number D, enhanced secretion of hydrochloric acid in stomach. Let me know A, B, C or D which of the following will be the correct answer for problem number 17. Again, I will wait for a minute. Then I will check the chat box. Which of the following activities occurred following the stimulation of H2 receptor? Vasodilation, uterine contraction, bronchial smooth muscle contraction, or enhanced secretion of hydrochloric acid in stomach? Which of the following activities occurred following the stimulation of H2 receptors? Let me check the chat box. Let's see if there is any answer in the chat box. Okay. Okay, I got one answer which says number C is the correct answer. Let me check if number C is the correct answer or not. The correct answer will be, it is number D, enhanced secretion of hydrochloric acid in stomach. It occurs with the stimulation of H2 receptors. So H1 and H2 receptors, these are the histamine receptors that couple with the GQ11 stimulating phospholipase C. The H1 receptors, H2 receptors, histamine receptors that interact with the Gs to activate the adenyl cyclase. The periphery of the body, adrenal medulla, vascular endothelium, heart and central nervous system is the location for H1 receptor, whereas the central nervous system, the parietal cells of the stomach is the location of H2 receptor. Antagonist diphenylhydramine, loratadine, cetirizine, clemizine, these are the antagonist of H1 receptor, and ranitidine, cimetidine, these are the for H2 receptor. Ileum contraction, vasodilation, bronchial contraction, these are the function for the H1 receptor, whereas the speed up the sinus rhythm, stimulation of gastric acid secretion, smooth muscle relaxation, these are the function of the H2 receptor. So H1 and H2 receptor, and H1 receptor is receptor that couples with GQ by 11 can stimulate the phospholipase C involved in the cellular migration, vasodilation and nociception, present in the endothelial cell, dermal cell, and on the skin, lymphocyte, dendritic cell, respiratory, epithelial cell, and nerves. H2 receptor is receptor that interacts with Gs to activate the adenyl cyclase, involved in the vascular permeability, gastric acid secretion, and airway mucus production, present in the smooth muscle cells, dendritic cells, gastric parietal cell, T cell, B cell, brain and cardiac tissues. Okay. Now let me go to the next problem. Problem number 18 says, which of the following heterocyclic rings is present in cimetidine? Your options are number A, imidazole, number B, pyrazole, number C, indole, and number D, benzimidazole. Which of the following heterocyclic rings is present in cimetidine? Imidazole, pyrazole, indole, or benzimidazole? Let me know A, B, C or D which of the following will be the correct answer for problem number 18. Which of the following heterocyclic rings is present in cimetidine? Imidazole, pyrazole, indole or benzimidazole? Again, I will wait for a minute, then I will check the chat box. Which of the following heterocyclic rings is present in cimetidine? Let me check the chat box. Let me see if there is any answer. Okay, I got two answers, number B, number D and number B. Let me check which of the following is the correct answer, number B or number D. The heterocyclic ring that is present in cimetidine will be, it is number A. Imidazole ring is present in cimetidine. So these are the examples of the aromatic heterocycles. Okay, so these are the 5-member rings, furan, pyrrole, and thiophene, which have a single heteroatom. Okay, then we have six-member rings like pyridine, indole, benzofuran. We have five-member rings with two heteroatoms like imidazole, oxazole, pyrazole. We have this two with two heteroatom. We have pyridazine, pyrimidine, and purine, for which are the two members. So these are the different heterocyclic rings. Okay, so this is the synthetic route of the cimetidine, which is originally carried out as a four-step process with the lithium aluminium hydride that is used as a reagent for the initial reduction step. Subsequent research revealed that this division could be carried out more cheaply and safely using sodium and liquid ammonia also. So it is used for the manufacturing process. So this first step is this reduction for this ester to hydroxyl group to the alcohol. Then this which can be done by lithium aluminium hydride or sodium ammonia and methanol ammonium chloride. Then followed by the reaction with this thiol, which gives us this amine kind of functional group. Then followed by this reaction, and which gives us ultimately the four-step reaction gives us a cimetidine. So as you can see, cimetidine has this imidazole ring, which is a five-member heterocyclic ring containing two nitrogen at the 1 and 3 position. Biological activity: cimetidine inhibits the gastric acid release by acting as an antagonist of H2 receptor. It does not show any side effect now observed in the for the metiamide, which is shown in the slightly more active. It has also been found to inhibit the pentagastrin from stimulating release of the gastric acid. Pentagastrin is an analog of the gastrin and is the fact cimetidine inhibits. It suggests that some relationship between the histamine and gastrin in the release of the gastric acid. Cimetidine was first marketed in the UK in 1976 under the trade name of Tegamet, and it was first really effective anti-ulcer drug doing away with the without surgery. For several years it was world's biggest selling prescribed product until it was pushed in the second place by ranitidine. Okay, so correct answer was imidazole. Now I will go to the next problem. Problem number 19 says which of the following statement is false regarding QSAR? Now your options are number A, QSAR, for QSAR study the compound studied need not to be structurally related. Number B, correct testing procedures must be used. Number C, in vitro tests carried out on the isolated enzymes are relevant. Number D, none of the above. Which of the following statement is false regarding QSAR? Your options are number A, for QSAR study, the compound studied need not to be structurally related. Number B, correct testing procedures must be used. Number C, in vitro tests carried out on isolated enzymes are relevant. And number D, none of the above. Which of the following is the correct answer for problem number 19? Read all the options carefully. Then let me know in the chat box that A, B, C or D, which of the following is correct answer for problem number 19. Again, I will wait for a minute, read all the options carefully, and then let me know which of the following statement is false regarding the QSR. Number A, for the QSR study, compound studied need not to be structurally related. Number B, correct testing procedures must be used. Number C, in vitro tests carried out on isolated enzymes are relevant. And number D, none of the above. Which of the following statements is false regarding QSR? Let me check the chat box because I think there will be an answer. Okay, I got one answer which saying number C is the correct answer. Let me check if number C is correct answer or is there any different answer. So the correct answer is number A. For QSR study, compounds are need not to be structurally related. This statement is false. Okay. So the QSAR approach attempts to identify and quantify the physicochemical properties of a drug to see whether any of these properties has an effect on the drug's biological activity. If such a relationship holds true, an equation can be drawn up which qualifies the relationship and allows the medicinal chemist to say with some confidence that the property or properties has an important role in pharmacokinetics or mechanism of action of the drug. This also allows the medicinal chemist to some level of prediction by quantifying the physicochemical properties. It should be possible to calculate the advance which biological activity of a novel analog might be. There is two advantages to this. Firstly, it allows to the medicinal chemist to target the efforts on the analog which should be improved activity and the cut down the number of analogs that had to be made. Secondly, if the analog is discovered which does not fit to the equation, it implies that some other feature is important and provides a lead for the further development. What is the physicochemical features we have mentioned? Essentially, it could be any structural, physical or chemical property of the drug. Clearly, any drug will have a large number of such properties, and it would be the Herculean task to quantify and relate them to biological property at the same time. A simple, more practical approach is to consider one or two physicochemical properties of the drug to vary this while attempting to keep the other properties constant. It is not as simple as it sounds, but it is not always possible to vary one property without affecting another. Nevertheless, it has been numerous examples whether the approaches work. It is important that QSR method is used properly and in relevant situations. Firstly, the compound studied must be structurally related, act at the same target and have some mechanism of action. Secondly, it is crucial to the correct testing procedures are used. In vitro tests carried out in the isolated enzymes are relevant for the QSR study as the activities measured for the different inhibitors are related directly to how each compound binds to the active site. In vivo tests carried out measured the physicochemical effects of the drug inhibitors are not valid. However, as pharmacodynamic and pharmacokinetic effects come to a play, this have the possible to derive a sensible QSR. So all the compounds that are studied with the QSR must be structurally related. Okay, and the in vitro studies on the carried out on isolated enzymes are measured in different inhibitors. Are it can happen, it can be taken, but in vivo test carried out the measured physiological property of enzyme inhibitor are not valid. However, okay. So this was the correct answer. That was number A. Now the last question of today's session, which says which of the following is not a QSR parameter? Your options are number A hydrophobicity, number B hydrophilicity, number C steric and number D electronic. Which of the following is not a QSR parameter? Hydrophobicity, hydrophilicity, steric or electronic? Quickly let me know because I don't have much time. Uh, Almost the time is over for our class, but let us go for another time. Let us just stay for 5 more minutes. Let me know which of the following is not a QSR parameter: hydrophobicity, hydrophilicity, steric, or electronic. Which of the following is not a QSR parameter? Let me check if there is any answer in the chat box. Okay, I got two answer, number B and number A. Let me check which of the following is the correct answer. The correct answer is, it is number B. Hydrophilicity is not a QSR parameter. So what is QSR? Quantitative structure activity relationship, or QSR is a mathematical relationship which correlates measurable and calculable molecular properties to some specific biological activity in terms of the equation. QSR attempts to find consistent relationship between the biological activity and molecular properties so that these rules can be used to evaluate the activity of new compounds. Understanding QSR regarding electronic effects, steric and lipophilicity. Physiochemical properties, hydrophobicity, steric parameter and electronic parameters are the parameters which are taken for care of by this QSR. The hydrophobicity by partition coefficient, pi substituent constant, steric parameter like Tufts constant and Varloop steric parameter, and electronic parameter Hammett constant and dipole moment. Okay. So many physical, structural, chemical properties have been studied by QSR approach, but the most common are hydrophobic, electronic and steric properties. This is because it is possible to quantify these effects. Hydrophobic properties can be easily quantified for complete molecules and for individual substituent. However, it is more difficult to quantify electronic and steric properties for the complete molecule. This is only really feasible for the individual substituent. Consequently, QSR studies on a variety of totally different structures are relatively rare and are limited to the studies on hydrophobicity. It is more common to find QSR studies being carried out on the compounds of the same general structure where substituent on the aromatic rings and accessible functional groups are varied. The QSR then consider how the hydrophobic, electronic and steric properties of the substituent affect the biological activity. The three most studied physiochemical properties are now considered So this we will not discuss it in detail. So remember, this hydrophilicity, hydrophobicity, steric and electronic parameters are most important for the QSA. So this was the last question of today's session. So today basically we have discussed about the several topics we have covered today. We have covered about anti-ulcer drug, we have covered about QSAR, we have covered about anti-cancer drugs, and also we have covered about cholesterol lowering drugs. So this is my email ID. So this was the last class for this NPTEL course of organic chemistry in biology and drug development. We will not have any regular classes from now on, but you can drop your queries via email. Also you can drop your queries via the discussion forum. Tomorrow we will have another last class, which is I mean the summary class of 1 hour from 4 to 5 p.m. That there I will summarize the topic. So if you have any doubt or query, please let me know before the class so that I can prepare accordingly. Okay, thank you all for joining. I will close the session for today, and the class PPT and the recording, it will be shared to you soon. Okay, so thank you all. I am closing the session.